Now I have everything in my front. So I will thank everybody before actually starting this presentation. So I thank Triple IT, DM Kanchipuram, Ministry of Education, Government of India, and the Malaviya Mission Teacher Training Center, my posted and Triple IT DM for giving me this opportunity to speak to my esteemed fellow uh, uh, colleagues about my area of research. I really thank them for giving me this opportunity. So, the topic of today's discussion is basically connecting the brain circuits to skill movement. We'll see each and every terms specifically when we go through the talk. So, it's a journey through motor control, learning, and action. So how this neuroscience, which are the circuits of the brain, you learn circuits in the brain as well as in the spinal cord. So, how do they evolve into an action, or how to actually enable us to perform day-to-day activity. So, what are the various stages? What are the various levels that you have in that process? We'll see the journey. So, that is one fascinating journey. That's what I feel when I first read these, when I first got trained and PhD in this domains. So, so first question is very simple. Have you ever thought about this? So we walk with our spine straight, or in some cases stoop, and we run and we pick and place things. Have you even thought about this? How are we able to do this? So how this task, which is a simple task, such as walking, running, or placing something, arranging something. So placing and arranging are different. So that is how the biological species actually is very well, no, distinguishes these activities. Now, both seem same, but both are different. Same, different, different. It's a showcase that when we see the presentation. So, have you ever thought this? How are we even doing this? So, those are gross body movements. If you see them, these are called gross body movements, and these are fine dexterous movements. So, those require not a fine control, but these movements, such as holding the tip of the fingers with a onto a zip, as well as onto a stitching needle and onto a pencil, or we actually placing it on a paper or placing it on a cloth to stick precisely, more accurately. How is this even possible? Have you ever thought this? The answer is very easy. The answer seems obvious. Answer seems obvious, but it's not obvious. So, how are we able to accomplish them? Yes, we are able to accomplish them with these facets, these structures that is actually available in our human body. So, what are these structures? So, one is the brain and spinal cord, which wires almost all the tissues in the body. In most of the cases, these tissues become muscles, and how the wiring actually takes place. What are the fundamental aspect of this brains and spinal cord, which is a neuron? So, brain and spinal cord together become a central nervous system, and this is the this is the central nervous system, brain and spinal cord. So what do they give us? They give us wires. So what are these? They're actually not wires, they're nerves. So these nerves, I'm just giving the analogy, we are engineers, so electrical, so wires. That's the reason we most of them we wire them. That's what we call in our field. So we have neurons. So these neurons actually take impulses from this brain, actually takes some small amount of uh, uh, you cannot call them a current, some electrical impulses. So that's the difference. So with electrical science, you call them as a current, because the flow in them is happening in terms of electrons, but here the flow is happening in terms of ions. Fundamental difference. So fundamental difference, that is how that's one of the reasons electrical current is bit fast, because electron size is smaller, but here ions are a bit larger. The ion size is larger because electrons is a part of that big atom. So you lose an electron from the atom, you become an ion. So so these conduct this electrical impulses in form of ions. So once these ions actually get this impulses, they are transmitted to muscle. So once the muscle actually contracts, so please be very clear, muscles can only contract, they can passively elongate. So that's a fundamental difference. So people usually say muscles will elongate, muscles will not elongate actively. Whenever I get an impulse from the brain, muscles contract. So once muscles contract, how muscles are connected to the bones? They are connected by tendons. So once you make a contraction of the muscles, the bones will move. So by this way, we can achieve our day-to-day activities. So that is how complex the system is. So I get some signal from the brain, and that some signal from the brain is actually transducted to the peripheral devices such as muscles and bones via your nerves, the fundamental units of the nerves or neurons. And when the nerves actually give the electrical impulses, they twitch. So that phenomenon is called twitch or contract. The smallest unit of a contraction is a twitch. So when that moves, your bones actually move. So all these muscles are connected to bones via tendons, but bones and bones are connected via ligaments. So walking, running, or pick and place, all these things actually do this big process. This process actually takes around like seconds. Voluntary movements, they take around 1 to 2 seconds in the execution pathway. So this is how these tasks were accomplished. Okay, let's quickly go to this neuroscience. So always credit the James, the father of neuroscience, Charles Scott Sherrington, who got Nobel Prize for giving a wonderful phenomenon called integrative action of nervous system. So he got this Nobel Prize in 1932 along with Edgar Douglas Adrian. So both were people are father as such as Sherrington. So he claims that this is a scientific study of nerves and especially how nerves affect learning and behavior. So that is the fundamental. So a circuit is formed, any circuit in the brain. So when I say circuit, a neuronal network, neural circuit is formed, and any circuit is not for just for namesake. They are formed for some specific reason. So how this specific nerve circuit is formed. It is to accomplish a task. So when you accomplish a task, we make it as a behavior. So we'll see what is this behavior and skill in upcoming slides. But have you ever understood this? Have you ever thought another day-to-day activity is we do this? So in the early morning, no, we want to check the shower if the water is hot or not. But in certain cases, if the water suddenly comes hot, let's say somebody has already opened the tap, suddenly comes, we retrieve the hand, right? So how the thing has this is take place? Answer everybody know that is a reflex action. But now only we saw something called volition pathway or voluntary pathway. So it comes from the brain, spinal cord, then it goes to the muscles, from the nerves, via ganglia, then to the skeletal, then something happens. It takes like 1 second or 2 seconds, but you don't have that much time when you are touching an odd entity. So what happens? Spinal cord immediately takes that action. See, this is how we should learn about teamwork. If you see both brain and spinal cord, they work simultaneously. But if, for example, I'm touching this hot water and I need to send the impulses to brain to actually process whether to retrieve the hand or not, my hand will be burned. So what happens is spinal cord, the system immediately goes to the spinal cord. Okay, it immediately reaches the spinal cord, and spinal cord produces a reflex action in milliseconds. So that is happening in milliseconds. Voluntary takes place in 1 to 2 seconds. So in within millisecond time, you are actually retrieving your hand. So this pathway is reflex. But also why there's better teamwork. So what happens is the spinal cord actually sends the motor execution plan, or the whatever the motor action that it has performed spinal cord, it will send a copy to the brain. That is most of the time you tend to realize after you retrieve the hand, the tap is on. That is most of the time you tend to know, you realize that the tap is hot after you have retrieved your hand. That is because the motor plan, the action executed is sent as a copy to the brain after you retrieve the hand, because the main reason is to save the hand from getting burnt, right? So then why do I need to send the copy to the brain? Very simple. So in future, I will not make this mistake. So that is how sophisticated your CNS is. That is how better we are as a living entity. Okay. Most of the living entities, as monkeys, rats, most of them have this kind of a phenomena. That is how good this spinal cord actually works. Okay, and you can see how they work. So you have like upper motor neurons, which is actually giving some circuits from connecting certain regions in the brain to the lower aspect. Then you have the spinal neurons, then you have lower motor neuron. So all these actually conduct via ions. This process of conducting the impulses via ions is called action. We'll see that. So this field that actually studies about nerves and how a circuit is generated in the brain is called neuroscience. For a specific task is called neuroscience. and Scott Sherrington is a father of integrative neuroscience. We'll see why the integration comes into play. You can see here why the integration comes into play. Here, you are taking the signal here, adding the signal to this, then you are adding back to the main lower motor neuron. It is actually summing up. It's basically a sum up. It's an integration system. So for that, find he got Nobel Prize, both admin and then comes so now you know how a circuit is in nerves circuits are formed, but there are also neural processes that actually plan a movement, initiate a movement, regulate a movement. Okay, so what are these process before you actually do a movement? So those side of things is called motor control. Okay, so people usually misalign this with controlling a movement during execution. No, no, no. This is actually controls that's existing in various levels in the CNS, neural processes that enables you to plan a movement, initiate a movement, and regulate a movement. It could be about voluntary, involuntary movement. So he's one of the founding fathers, Nikolai Weinstein. So he's a founding father. So he wrote all this wonderful theory in his book. So one of first and foremost people to actually start with this field called motor control, Nikolai Bernstein, and then everybody might know Fitz. So Fitz law. So for a rapid air movement, you can actually use the length and the width of the movement to actually see whether the movement is achieved accurately or not. That's Fitz law. So these both of them, they come up with a strategy to say how the movement is actually formed to execute. But the problem that our CNS as it will most of the time it encounters this degree of freedom problem. That is, so let's say this is a goldsmith, is a goldsmith, and this goldsmith is going to hit the hammer on the plate every time. Every time he hits in a different place. He's an expert, still he hits in a different place. So that is called motor variability. So this is called motor variability. And why you got motor variability? Because you have abundance in your system. So this abundance is called redundancy. So how do I say? So this is a 3D point in space. This is a 3D point in space, and I like 7 degrees, more than 7 degrees. So the three equations, seven unknowns will refine in an infinite solutions. So many set of equivalent solutions. So this motor redundancy and variability is a daunting challenge for CNS. So how the CNS actually then produces the movement, initiate and execute them becomes the control aspect of your motor system. So we call this as a motor system because the data is coming from the brain to the peripheral organs. We call them as motor neurons, motor pathway, motor neurons, motor neurons. So this is motor control. So precisely neuroscience, we study about the circuits, motor control. We study about how movement is planned, initiated, regulated for via various structures in the brain. Motor cortex, cerebrum, then you have cerebellum, basal ganglia, and you have your spinal cord. Okay. So mostly spinal and pons are for involuntary movements. Then you have something. So once I know I'm going to the movement is actually planned, then what we have is motor learning. So this is another important aspect. So if you see, so he's termed as a father of modern motor learning. So if you read this definition claimed by Skimmett and Lee in one of his books, so the set of processes associated with practice, so and experience leading to relatively permanent changes in the skill performance. We'll see all these things. So what I do. So I now what I have is a movement that's being generated, but is it correct or not? So I have a goal to achieve. So what are those tasks we saw? Walking, running, picking, and pacing. So if you see these goals, are they reached correctly? Are they executed correctly? So I should not fall down when I walk or run, and I should place the items correctly. Correct. So for that you need practice. Do the same thing repeatedly. Practice does not make perfect. Practice improves the performance. That's what Rosenbaum in his commentary famously quoted in 1900s. So it's always not practice makes perfect. It's the practice always improves your performance. There's always no perfect. Every time you increase the performance, it might be a big increase or a small increase. So what happens is, so I do, let's say, no, I do a task, walking, talking, running, all these are motor tasks. So these tasks actually gets stored in the memory. That's what it means. So there are two memories. One is working memory, another one is permanent memory. So when we are doing a task on a very first day, it stores in the working memory. And when we sleep, there's something called offline consolidation takes place. So if the brain actually sees this task is performed on a day-to-day basis every day, then it goes and sits in the final permanent memory. That's the hippocampus and amygdala, right? So they actually form a neural circuit for that task. So now you have developed a skill. Now you have developed a specific pattern, a specific way to perform a task that becomes your behavior, and behavior that execute that actually ends up in a 100% execution of a task. Successful completion is called skilled behavior or skill. So when do I achieve a skill? With practice. So what happens when I do practice? Neural circuits in the brain changes. They actually changes based on the skill that we are developing. So and when I want to use this skill, for example, I know driving very well. I've driven like more kilometers, 5 or 10 lakh kilometers, let's say. Uh, Suddenly I go to suddenly I'm not driving for a quite long period, like 2 to 3 years. My skill is not it won't get wasted. Even after 5 years you go, the skill will remain. But always there is always a point where the skill would actually needs more practice to revive that. So you need to unlearn or relearn. So these are phenomena in motor learning. So as such with motor learning, now we have a moment. So how do you practice this moment? How many times you practice the moment? So cricket players they practice many times to become skilled in that field. How many times that field is called motor learning. Then you have this motor action. Famous personalities out there. So one is My Bridge. So you can see this person walking. So he actually used photographic plates in the 1800s. So actually capture these moments. He wants to see what is the distance. So you can see the small square, big square in the image. So these are photographic plates. So they will actually identify the distance between, just like how we do pixel analysis here. So distance between the stands, how quick they are moving. So my bridge came up with this phenomenal experiment with male, female uh, people walking on a platform with horses, with so many different animals. He actually quantified first person to quantify uh, image movement analysis was my bridge. This is not a camera. This is actually a big bulb that will break when you actually take a photograph. So like that, no, each bulb was costly at that time. It's equivalent to a cost of a projector bulb that time. So that is how tough it was. So he captured this. So he told these movements could be quantified. So what is a motor action? So I know I have a movement that is planned, initiated, regulated, and when it's coming as an output, they become motor action. So that is the difference. So you have your motor control where we plan, regulate, initiate a movement. So when it's actually done by the peripheral system, legs, hands, the entire trunk of the body, head, they become motor action. So how good you do the motor action, you need motor learning. So these are the phenomena, and initial this is for this kind of movement quantification we call as kinematics, because geometry you can see is actually moving something like on a graph, so from one coordinate to another coordinate. So these are geometry of movements. So geometry of movements are kinematics, where you will actually quantify the displacement, distance, position, velocity, acceleration, and jerk, and quantify how the person is actually making a movement. And then this is kinematics. And also the French civil engineer Regnier actually provided something initial force sensors. This is a dynamometer. So so he wants to know. So what was the reason is he wants to find what is the strength of the muscle. So he used this. You can see there are like 2 points. So he used two hands of the person to grab it and press it. So there are like based on the movement in the scale, you can say the strength of the muscle. Similarly, you can pull it as well. So two things he wants to. One is the compression force, another one is the tensile force. So he was one. These two people are one of the few people in the early, not even 1900s, in the early 1800s, they came up with the wonderful tools. wonderful, almost accurate tools to quantify the movements. Why was now you are immediately saying quantify movements? Yes, I need to know how. So this movement quantification. So why these people are actually walking or running? That will tell whether the person is healthy or not, whether the person is actually elite or not. Like either he possesses a skill greater than a normal person as an athlete. So two spectrums we can able to see. That is why we quantify these movements. So then, before even Newton wrote his force Newton laws, our Giovanni Alfonso Borelli, the father of biomechanics, actually studied this muscular contractions. So he told this muscular contractions are actually will give you a force. So before to this, we saw a person quantifying force, but before to all this, even before Newton wrote his first law. So this is an interesting fact. That is why this person, no, the Giovanni Alfonso Berli is considered as the father of biomechanics. He told human bodily movements are caused by muscular contraction. So bodily movements. So we are yet to work on forces here, but that's what it means. Bodily movements. There are two things bodily movements could actually be seen. One is force, another one is kinematics. One is kinetics, another one is kinematics. So he told human bodily movements are caused by muscular contraction way before Newton. So father of biomechanics. He's considered as the father of biomechanics, and this is his first the manifesto on the motor animalium. So where he actually went through so many animals and human phenomena for his study. So that is regarding biomechanics. So we have seen like four different things about our area. So one is neuroscience, motor control, motor learning, uh, then biomechanics, and finally rehabilitation we'll see last. But before to that, we'll quickly understand, we'll quickly see how uh, this neuroscience as such as a field is what they want and what are the basic fundamental aspects of our neuroscience. So neuroscience, as I told, they study this is a field that actually addresses the circuits in the brain, the anatomical physiological circuits as well as the major circuits that is enabling control coordination of various movements of body, various movements in the body. Everything is movement as such, as a movement scientist. I always claim it, just like anybody. Everything is movement. Everything there is somewhere something is moving. So that is what it differentiates. Differentiates an organism, a cell, whether it is alive or not. So the brain, as such, if you see, there are two things in neuroscience. We study the brain and spinal cord. How the circuits in the brain are working, how the circuits in the brain, spinal cord are working. So first, let's say cerebral cortex. So we'll go through one by one. So if you see, this is a cerebral cortex. So this is where most of your lobes exist, which is your decision-making lobe, the frontal, the motor cortex, where the all voluntary movement. I told you, know. So the neuron has to travel from your finger to the through the spinal cord, then to the thalamus, then to the brainstem, to the thalamus, then it gets related to cerebral cortex. So it is that much time it takes before that is what we saw. If you don't respond upfront, your fingers will be burned. So that is why it takes 1 to 2 seconds for voluntary movement to actually. So I want to move from this place to another. I want to join this meeting. All are voluntary movements. So all these takes like 1 to 2 seconds. So I decide here, then I plan my decide, plan, everything happens. We have a prefrontal cortex before emotion region. So that planning and initiation takes place. Then we actually make sure which are the peripheral that needs to be given command. We give command, then we move. And based on that, the feedback mechanism works. So here, if you see, we have something called basal nuclei. So these basal nuclei are movement initiators. So the cerebral cortex will actually give signals to basal nuclei. Tell initiate these leads movements. Then I initiate the movements. Correct. So once I initiate the movements, I always correct the movements. Let's say there is a so I'm walking. There is a oil on the floor. I should not slip. So I will walk slowly, I will walk carefully, I will make sure I maintain my CG, center of gravity. So that thing is taken care by cerebral movement correction facility. That is why people when they alcohol themselves, the alcohol molecules pass the blood brain barrier and affect the cerebellum. That's why they will have this shaky movements on both sides of the body. And these guys, on the other hand, controls the involuntary movement. Thalamus and hypothalamus, they do various functions. One of the important aspects of thalamus is relay. So that is why when something happens here, we don't get the signals from the cerebral cortex. That is one of the reasons. We'll see what is that something when we go through rehabilitation. Then you have spinal cord. So you have like various sections of the spinal cord: sacral, lumbar, thoracic, and cervical. So each of them providing the nerves and other aspects for various different sites of the body. And you can see that this is how the vertebra of the spinal cord looks from the lateral side, not from the lateral, from the anterior, from the posterior side. So you have like different matters. And so spinal cord, as of now, let's have this into our consideration. So I have cerebral cortex for voluntary, I have basal ganglia for initiation, cerebellum for correction, while I have spinal cord for reflex and other functionalities. Let's have this for understanding until now. So why is this? Okay, we are electrical engineers. We are not core biological people. So we need to know what is really happening. So very simple thing that we have learned in our 12th standard is V equal to I R, Ohm's law. So wires, that's what I was telling. Now we are seeing about central nervous system wires. What are these wires? So these wires actually take the electrons or the current. The flow of electrons is the current. When you put them in appropriate voltage, that is why this voltage is pushing this current. Then if there is if based on the gauge of the wire, the material, area, length, there will be resistance that will actually restrict the flow. Okay, so this is exactly the same with neuron, right? So these are various different forms of Ohm's law with respect to power, voltage, and resistance. So same thing is exactly the issue with your uh, biological system. But the important thing is here you have ions. So I don't have electrons here, I have ions. That is the reason you need sensors and transducers. So sensors, when you keep an ECG electrode, what it actually does, it actually going to convert your ionic current into electronic current. So that's why it's called a transducer. So that is how important that is. So designing that itself, that's one of the areas that I work. So ions, as such, if you see, these are the fundamental ions: sodium ion, chloride ion, magnesium, calcium, potassium. These ions are available in the electrolytic body. So based on their proportion inside and outside the cell, the value of voltages changes in the cell. So let's say, we saw that shower example. We saw that shower example. So initially, the cell will be in the resting condition. So any cell, neuron, cardiac cell, or filament in muscle, we don't call it as a cell, we call it as a filament because it is elongated. So any cell you take in the body of any tissue, so you will have a cell. So once that cell is getting a stimulus, the heat is a stimulus. In that case, then what happens? The heat sensitive channels. So we have channels. So as such, okay, let's quickly understand what is a cell. Cell is actually separated from the extracellular fluid via membrane called semi-permeable membrane. So that is permeable only to ions. It's permeable only at certain places called ion channels. So we have something called ion channels, and these ions move in and outside the cell via ion channels. Rest are hydrophilic or hydrophobic. Rest of the cell wall. These things we know. So when the ions actually move inside and outside the cell, sodium ion comes inside. So initially there will be minus 70. When sodium ion comes inside, they actually yeah, reduce the negativity once the threshold. This is the gate threshold. This is minus 55 for normal cells. They immediately produce this all or none phenomena called action potential. So this is this all or none phenomena where this phase is called depolarization. That is making the cell to more positive. You can see it has reached to 30 millivolt. The cell, if you put two microelectrodes inside and outside the cell, we can find this value. It has reached 30 millivolt. Then repolarization, where the voltage, where the sodium potassium gates open, voltage gated potassium gates open, because that is the threshold for the potassium gates. Once potassium gets open, the repolarization actually happens. Then once in the repolarization phase, the sodium pumps actually happens in the hyperpolarization, because it has reached below minus 90 or minus 100, but we need to bring it to minus 70 millivolts. So this phase actually takes place. So this is what we call it as spike all or none phenomenon action potential. So whenever a cell is actually having this, so this is this information from external world. What is the external world? It could be hot, heat, anything. So this is how a signal is getting transmitted in your neuron. So you have this signal. So what is happening? As I told you, you have semi-permeable membrane, you have activation gates. So they will be open when the threshold is reached. So ions will move out. If the activation gates are closed, the ions cannot move out. So based on this, any cell will be activated or deactivated, or we call it in more biological way depolarized or depolarized, polarized. So initial resting is polarized state where it is always in negative state. Then in depolarized, it becomes positive. Then it becomes repolarized and it reaches the resting state. So this is all or none phenomena. Very important thing is here we have two things. One is the absolute refractive period, that is from 3 to let's say, no, you have like 3 to 4. Before this, you can see my pointer, this place, you can see it as absolute refractive period, or I guess at this point, until this point, you cannot do even an extra stimulus comes, this is not going to change the state. But when it goes to the refractive period, you give a more bigger stimulus, on another action potential is going to be produced. So what it does, so I'm going to give, there is a bigger stimulus. What is the stimulus? Environmental stimulus, heat, chemical, something is bigger is happening. So I need to inform the central nervous system. So immediately the relative refractive rate produces another spike. So based on this frequency of the spike and based on the level of number of spikes, you can actually modulate the signal, modulate the information. That is, information is transmitted to the central nervous system from the peripheral organ. And so from the both vice versa, from the central to peripheral as well as from peripheral to central, how the action potential is conducted. This is how conducted. So neuron as such is this is the basic cell in the nervous system, that is neuron. So you have a cell body, you have nucleus, you have the cell body because we have dendrites here, and these are the dendrites that connect to other neurons. Then you have axon. So these axons are myelinated sheets. So the main reason of myelinated sheet is you can see this wire. This, so just like that's why I told this is a wire. You have a wire uninsulated, then you get a shock. You lose electrons because the electrons is passing through your body. Correct. Same thing. These are this sheet, myelinated sheets are insulations, so that you don't lose this action potential that is coming. So what happens is action potential will actually takes place at this junctures called nodes of Ranvier. So where the action potential changes here, so there is a spike here. Then it quickly goes here. So it looks like it's jumping from this point to this point called saltatory conduction. So this is how quick the nerves will actually conduct the impulse. So because that has to respond very quickly, 1 to 2 seconds is also very quick. Like milliseconds is quicker, but 1 to 2 seconds is still quick. Okay, so this is how the information is transacted through the body, from the periphery to central, from the central to periphery. So you can see these things. Sodium ions come inside, make them positive. That is depolarization. Then once the impulse is moved, they repolarize. Then they go to resting state. So once they reach the tip, so this was the Nobel Prize. This is why Sherrington was awarded the Nobel Prize. We'll see that when we reach when we see that we'll see. So what is why he was awarded the Nobel Prize was he was able to find there is something in the lower part of our neuron that's actually making summations. Summation processes just like in a regular mathematics, integrative action that's called a synapse. So for his work in that direction, he got Nobel Prize. So until this point of our neuron is conducting, so now our impulse has reached a synapse. We'll stop here because I continue the discussion when we reach to motor control. Because from once it reaches the synapse, it has to go somewhere. Either it can go to another neuron, we call it as postsynaptic neuron or presynaptic neuron. So that is here. So this is a presynaptic. I have another neuron that's connected. It's called a postsynaptic neuron. Either it goes to neuron transmission or it can do neuron muscular transmission because that is what we are interested in, right? So I have a signal that has to pass through the neuron to the muscles. So we'll see that. So before to that, we have something called fMRI, which is functional magnetic resonance imaging. So why this is important? Because this is one of the areas that I recently worked enormously. So what we do is we actually give them a function task. So when they do a task, we are taking MRI. So functional magnetic resonance imaging. Why we go for this MRI? Because it has a wonderful spatial resolution. I know from which point what is happening. So we call this as activation. We'll see that in the preceding slides. So this fMRI primarily depends on bold signal, which is oxygenation level dependent signal, which is an indirect measurement. So what happens is you have a neural activity. So I am doing a task and making a cognitive calculation in my brain. So neural activity increases, blood flow increases. Whenever I say blood flow increases, oxyhemoglobin increases, which is going to improve my MR signal or T2 weighted image or MR signal. So by this way, I could actually isolate that region of the brain. So that stimulus is something, a task that I'm doing. So because of that, there's a neural activity. So when there is a neural activity, there is a so what happens when a signal reaches the let me see if I have any questions in the background. Okay, so no more questions. You can ask me. You can stop me at any point, and you can ask me. So so whenever there is a signal that's going to reach the end of a neuron at the synapse, it's going to give something called neurotransmitters. That neurotransmitters is going to talk with other neurons. So there are other coupling. This is neurovascular coupling. Neurovascular coupling is different. This is a neuronal transmitter molecular coupling. There is a neuronal coupling that takes place, but most of the cases we have a transmitter. So whenever electric impulse reaches the end of the neuron, it gives transmitter. This transmitter actually sits in the dendrites or the vascular channel or the I mean ion channels in the other neurons and transduct the impulse. We'll see that in some time. So whenever I have neuronal activity, there is as I told you, there will be a hemodynamic response. There is improved activity in the blood oxygen level. You all know MRI oxygenation signal is pretty much a detected signal. So once you have this detected oxygenated signal, we have this thing. We have like echo time spin gradient and all these things based on magnetic field strength and all these things. We can determine our bold response, which is the fMRI image. Okay, so before going to this, so this is the difference between you have like one. So whenever I have MRI as such, I'll be having a very high clarity, high resolution, spatially the system. I'll be having one image. But when I go for fMRI, I have different slices of image for every 2 seconds for 5 minutes. All are having low resolution, but they have a very good dynamic response. That means the functional aspect, at which point, a specific, which point in the brain a specific functionality is taking place. Let's say, I'm seeing occipital lobe, I'm thinking frontal lobe, I'm making a plan prefrontal cortex. So those things will be highlighted. We call this highlighted as activated or activation function. So how do we calculate this activation function? Very simple. Once we stimulate with the task, then before stimulating a task, there is an image MRI. After stimulating the task, there is MRI. We subtract them, and then we'll get as a difference. So this difference is the activated region in the brain. So individual images, we take different slices and take mean difference image. So that image only we are seeing as fMRI, and these are different planes of images or slices. One is a slice that's going to differentiate top and down, axial, front and back coronal, left and right sagittal, and these are certain images or networks which we are pretty much important for biomedical image processing engineers to actually go through. So if you see, we have something called default mode, visual mode, sensory mode, motor, salience, dorsal. These are networks. So these are networks that gets activated or highlighted in an fMRI image when a specific functionality is taking place. For example, if you say default mode, which is ventromedial prefrontal cortex, then precuneus posterior cingulate, bilateral inferior parietal regions. So these are the regions that is actually getting highlighted, and when they are getting highlighted, when there is an episodic memory that's taking place into account. Similarly, your occipital lobe will be working when there's a visual stimulus. and sensory motor for the primary sensory motor. When you get sensations and appropriate response, you get primary sensory motor cortex, sublimated motor area are getting activated. Similarly, when you go for executive, so these are controls above your cognitive functions, complex cognitive functions such as planning a game, executing a game, and changing the outcome of a game. So higher level cognitive functions we call as executive control. So, what are those areas? Which is mesofrontal regions, then dorsolateral frontal areas, then your parietal cortex. So, when they're having working memory in attentive functions, so those things, these images, these networks actually enable us to. If you go to auditory temporal lobes, so we'll see that. So this once we complete this, we'll see my work in schizophrenia, where the temporal lobe actually plays a wonderful. So these networks, default mode network, as well as dorsal attention network, plays an important role. So attention and controlling demands. You have this dorsal attention networks in fMRI. Default the same episodic memory. See, this is memory, and these are controlling demands, and these are for auditory. Just have this in your mind. I'll show you some. With schizophrenia. So this schizophrenia work got published in 2024. So I started working in this after seeing the complexity of this work. So I collaborated with one of the faculties in computer science, where the faculty told to use some sophisticated deep learning method. Which is called Graph Sage and GCN, Graph Convoluted Neural Network. So the main aim is to actually identify why the schizophrenia people get audio-verbal hallucinations. So when I say audio-verbal hallucinations, it is actually a verbal phenomena. They tend to hear without any stimulus. So they get these hallucinations quite frequently. So, but still, people are having challenges of finding which area of why these circuits are getting affected. Then, actually, I went through one of the papers, and I was able to see they were not able to find any differences. So, this was this paper that where you can see they are like different groups. One is healthy control, schizophrenia, and in the schizophrenia group, you had like audio-verbal hallucinations or no audio-verbal hallucinations. So what we did is we once we have this group, all the fMRI images, we actually constructed the graph. We constructed the graph, and then we actually went on to make a graph stage convolution model. So we were able to. So what is the and these are the metrics people we usually know, the standard metrics. So so what is the test that we were able to do is, so there are like different tests that was imposed on them. One is sentences, another one is words, another one is no sentences. So there will be a stimulus, sentences, words, and no sentences given to both hallucinating and non-hallucinating patients. So when that is given, fMRI was taken. So so what happens is, so temporal lobe, as I told you previously, you can see here whenever you hear something, temporal lobe, and when you have so this guy gets deactivated in goal-based attention tasks. So I need to listen. So goal-based attention task. What is that? I need to listen to a sentence, word or no sentence, right? So this mode gets deactivated. See, that is why. And this is one of the important aspects of by default. So only when you have goal-directed tasks, this mode should be deactivated, right? But in schizophrenic patients, by default, they will be deactivated. That is why they have this fragile, they don't have this attention, they have this fragile memory, they have this hallucinations, and they have this fragmented, as a fragile, it's fragmented memory. It is because of this reason they have that. And if you see this dorsal attention is also gets problem. That is why it's not easy to get their concentration back on a task. But this guy, the auditory will all temporal lobes will always be activated. So what we were able to see, so initial previous paper result was very simple. They told there's no difference between both this thing, and temporal lobe was the temporal lobe was not having. Yeah, I mean, they were not able to find previous paper. The paper before our work actually showed for any of the conditions, words, sentences, reversed conditions. So that is one of the reasons. If you see in movies, no people, if you have seen famous Exorcist movie, they can actually say a sentence the reverse direction. That's because of this phenomenon. So what happens is we also have given them a cue in reverse sentences. So there was no difference in activation for these any of them. So that tells that temporal lobe is actually not the cause for this hallucination. Rather than it's just merely a competing entity. But our graph, our work actually showed that it's not only temporal circuit, there's also a prefrontal circuit that's involved into the schizophrenic patients. That's what we were able to identify. These were able to clearly classify between this the small difference. So until this point, with other models, we are not able to tease out that small difference. But with this model that we just used, we are able to tease out the small difference that highlighted that it is not the stimulus that is actually competing. This is actually another circuit that's actually causing this hallucination. Uh, so that is why this research is pretty much important, right? So then let's quickly go. So now we have seen uh, how a neuron actually conducts impulses and how they are ready to give an impulse as an outcome. Now we need to take this impulse into a muscle. So we have seen a work how to quantify these neural circuits using fMRI. Now let's see how the impulses are transducted into these muscle fibers. So muscle fibers are action. They are motors. They are the biological, natural motors available in our body. So how are you going to give current to these motors? Obviously, by a neural system. And this is the cross sectional view of your muscle. So this is a bulk muscle, and you have each of muscle cell is called muscle filament, and inside the muscle filament you call myofibrils. And these myofibrils are actin and myosin filaments, and these actin and myosin filaments need to contract to produce a force. Very important. So this force is what is enabling you to produce a movement in a day-to-day activity. Lift the hand, uh, walking, placing pick and place, and so many. What is it? Writing, stitching, zipping, all fine dexterous movement. This actin and myosin filaments are enabling us to do. And how these actin and myosin filaments are conducting, we'll see. So this is your motor neurons. These are the synapses, and we call something called neuromuscular cleft, or this is the neuromuscular junction. And so action potential comes here. So you can see these are myofilaments. So the myofilament, a single myofilament. So you have many myofilaments form a filament, and each filament can produce a piconewton of force. That's how small it is. So you have millions of myofilaments and millions of them, then you can see how many newtons. You know, 1 Newton is 9.8 into uh, 9.8 meter per second squared into the appropriate weight, that is F equal to W into G. So that is one to produce 1 Newton, so many myofilaments need to be recruited. That is where the recruiting principle comes into play. So whenever I say recruit, these motor neurons are going to send the electrical impulses to these muscle filaments. And how am I going to do this? So this is the synaptic cleft, and this is your muscle filament region. And once the impulse reaches the synapse, there is vesicles that has neurotransmitter that will be released, and these neurotransmitters will sit on the neurotransmitter or ligand based ion channels in the muscle filament, and that will allow the sodium. So as I told you, once sodium enters, the muscle becomes positive. So once muscle becomes positive, they trigger the calcium. That is why the calcium is pretty much important for us. It's not only for bones but also for contraction of muscles. The calcium is important. Without calcium, the muscle cannot contract. That is an important aspect, because the calcium binds to some things in. So we have something called, as I told you, actin myosin filament, which acts as a ratchet kind of a mechanism. So this is one myosin end. It actually fits into a hole. So this is actin, and it fits into an hole. Actually, these are troponin tropomyosin complex. So in resting position, these holes will be filled by these complexes. So in excited state, they will attach to these holes and pull the muscle filament. So this is how they pull. You can see here, it is attached, it pulls, and it detaches for the next cycle. And so for the myosin head, it needs It needs power. It needs ATP. So the ATP is converted to ADP and phosphate. That bond is broken to give energy. So once that so you have your neurotransmitter. Acetylcholine, which is actually sitting on the postsynaptic membrane, that is your muscle filaments. Sodium enters, sodium channel opens, the impulse actually starts. These are voltage gated calcium channels. Calcium in sarcoplasmic reticulum. So, what is this sarcoplasmic reticulum? There's a small thread-like networks are called reticulum. They are the storage houses of calcium ions. That is why when you have a lesser calcium supplement or no calcium, calcium is taken from the bones. That is why bones become calcium deposits erodes from the bones, deposits into the blood, and it's taken to this muscle filament to make contraction. Because without calcium, there is no contraction. Without contraction, we cannot move. So that is why muscle bones become weaker. So that is why we should take calcium. So calcium is pretty much important. So once why calcium is required, because the calcium is going to flip this active myosin to show the holes to the myosin end. Without that, the troponin and tropomyosin complex is going to not show them. So this is called a sliding filament model. This is one of the phenomenal model that was explained about how muscles even contract. Very clearly understand muscles can contract. Okay, they can passively elongate. They can passively elongate, but they can contract to produce force. So what is that? It's an order of piconewton into 10 minus 12 Newton, right? So that is how small Newton a myofilament which can actually produce. So what is a twitch? So a twitch is a single action potential that reaches the muscle fiber that produces a unitary contraction, one directional contraction, and this twitch lasts for few milliseconds to 100 milliseconds. The duration of this action potential is 10 milliseconds. Also, there is always a gap between the electrical signal reaching and the mechanical system making a movement. So that there is always a gap. Hence, the mechanical effects last more than the electrical effect. That is the reason. So, if you see, this is the muscle, this is the tendon. So, this muscle is twitching, that means contracting. So, it is shortening. See, contracting and shortening. It's able to pull this. It is able to pull the bone. And in this, it is called isometric. So, this is called isotonic because I have a load, a constant load that I'm pulling. And here, it's called isometric because the length of the muscle remains same. Another immediate question people usually ask is length of the muscle is same. How can you call it as contraction? The muscle is actually contracting it, but the tendon and muscle length, the combination is not. It is actually remains same. That is why it's called isometric. And what are these cases when I'm holding a immobile object, I try to pull, I standing, sitting, all these things are isometric contractions, different types of contractions. So contractions, I have force, very important. So this is what an action potential reaches after a latent period. After that point of period, a force is produced. So this force is enabling us to do movements. So very important. And immediately, I have like different action potential coming. The force is actually trying to increase and results in smooth. That means repeated contraction of muscles, which is always tetanus. This is also caused by a bacterium. Due to the tetanus is a very important thing, and tetani is a convoluted method of production due to the several impulses that is getting reached. And this is how you modulate the force. This means this is how you modulate so action potential. So whenever I say this guy, this is actually coming from the central nervous system, the electrical impulse action potential. And this is the mechanical effect. This is the electrical effect. This is the mechanical effect. So you produce a force when there is a electrical impulse. And so by this, people came up with various different models of providing a mechanical understanding of a muscle. So initially they thought it is a spring, but it is also having some restrictions, right? So they put a damper, but it also needs some energy. So they put a power supply. So this is a so one is actual force that is getting generated, that recruitment, the power that you saw, the power that is getting generated in ATP for ratchet for pulling the actin myosin end. So that is this power. Then you have this two springs with a damper, restriction to flow. There is always a restriction to movement. It is not a fluid of a seamless flow. There's always a restriction to movement. So with this, we can actually put one as a velocity component of the force, another one is a displacement component, another one is an acceleration component. So the force is now mapped to the velocity, or the force that is produced is now mapped to the displacements of its anatomical units. Very, very important result. This is a very, very important. This set of results will lead us into Fitz law. This set of results will lead us to various different aspects of biomechanics. So very important result in biomechanics. Now we have reached the stage of biomechanics, and very this is another curve. We always tend to misinterpret this curve. So you see tension. So this is y-axis is tension, x-axis the sarcomere length. So when the length increases, something is happening. Very clearly, you see the length will increase only after a certain point, only until when all the, so this is called overlapping region. This is actin and this is myosin. This, sorry, this is actin and this is myosin head. So if all are actually connected to this, all the heads are actually connected, the actually the tension increases. You know, all are connected, so tension increases. So I can lift more space. Okay, but you can see there is no big change in the sarcomere. So okay, before to that, this combination of actin and myosin we call as sarcomere. Okay, sorry, this we call this as okay. Various sets of these cross bridges we call as sarcomere. Okay, I missed it. So without telling that, I cannot just all we can have it as muscle length. For a group discussion, we can have it as a muscle length, and actin and myosin molecule together form a cross bridge. Whether they are connected, only they are connected. If they are overlapping, the muscle force will be very low. If they are connected to all the actin myosin, it can produce more. It can actually tend more force. Then, if it is moving apart, this is after elongation. So I have a weight. After certain period, I cannot hold the weight because it has gone to absolute refractive period. So there is no weight. So I cannot. So what happens is I cannot lift this weight. So force required to lift the weight goes down. That's why I'm not able to lift it. But sarcomere length is increasing. Okay, so when force reduces, sarcomere length increases. That means there's no cross bridges. This is called passive elongation. So what happens? So what happens from the brain? You want to, so your muscle is as such. It has gone to a fatigue or a tired state. It has reached absolute refractive period. But so that is why you have a passive tension at a point called equilibrium length, or we call this as a resting length or equilibrium length. But suddenly you wanted to increase it, so you actively recruit from this point. What is active recruit? So I intentionally recruit more motor neurons to produce force. So this phenomena at this point is it will enable you to still lift. So I'm tired, my muscles are all tired, but still I can still lift because I can actively recruit other motor neurons. So whenever I say motor units or motor neurons, it is the connections of motor nerves, motor neurons with different filaments. So different filaments have different motor units. So different motor units will get recruited. This is how they control the force. The force controlling from the central nervous system happens by this level. So this is called Eneman principle or size principle. So this is very deep into biomechanics. So then you can see, so initially, I'm actually recruiting, I'm actually contracting, the force is increasing. Then after a certain period, I'm not able to lift this. I'm just, let's say I'm holding this weight for quite a period in time. So I'm totally tired, the muscle is totally tired, so it's going to elongate. It's going to drop due to elasticity of the muscle. We saw elasticity, the spring nature. It is going to drop. The tension in the muscle is going to drop. But but I am actually trying to do certain things from here. So I'm going to lift my shoulder, lift my other muscles here, and then try to lift this. Still, I can produce total tension. Okay, so this is called active recruitment after the resting. So what we strategize is in control theory, in motor control theory, what we strategize is very simple. So we have this set of values, set of, so we change these gains so that you can actually recruit different muscles for different functionalities. That is why when you are sprinting, separate set of muscles will be so for sprints. People who are going to do a sprint, they have a the easily fatigable muscle. The people who are going to go for marathon, they are going to have a long fatigable muscle. So that is how the central nervous system actually recruits. That is how the recruitment pattern actually takes place. So we have like large motor neurons, smaller motor neurons. Initially, I need a large force, I go for large motor neurons. So there's a spike, but they will take time to restore energy, right? So then you go for, but I need a large motor neuron, and then I need to stay. Then I go for recruiting short motor neuron. So this is recruitment pattern of the central nervous system, so that you will produce appropriate force for your task. Okay, so this is where the controlling aspects comes into play. Okay, then so until that point you have your controlling aspects. So that is, so I need to recruit a specific set of muscle filaments or a specific set of motor units for to produce a specific set of force. I need a constant force, I need a dipped force, I'm actually recruiting. This is a control strategy. Now I'm going to do the same thing, practice the same thing again and again. Okay, so let's say how practice improves the performance. So let's take a simple task. Let's say no. So this task was performed by Sosnick on principal investigators in pioneer groups of motor control labs. So this person actually, what he did is, so he had a tablet, so which has X and Y. It's a 2D tablet, and the purpose of this task is to move from different points on the screen. So initial days, he moved. There were like four discrete bell-shaped profiles. So this is a velocity profile. So I have a, I'm moving from one point to another. So this is, I'm moving from one point to, so initially I will be in one point, moving from one point. So this is a 2D, so this is a 2D movement. This is a 3D movement. Okay, this is what the daunting issue of central nervous system or the degrees of freedom problem or goals with mainstream problem. This is the that problem. Okay, so I'm going to move from one point to another in the space. This is a position movement. So when I'm going to take the displacement of the position, I'll be having like four the individual velocity profile. That means there are four stopping points. Initial days of practice, I'll be moving A to B, B to C, C to D, and D to E. So there are like four stoppage points. But with practice, I'm going to combine this stopping points. So initial, then I'll be having at only two stopping points. Then after I'll be having only two and four I reduced with 5 days of practice. So with practice, so so discrete movements are not always. So these are like gaps. So these are like discrete movements, just like how we stammer and speak, how we disconnectedly speak. Now we can speak continuously. That is always very good. That takes lesser time, less effort, but the outcome is also very better, right? So same thing. So I need a smooth, continuous movement. It takes practice. So this is how you quantify the kinematic this thing. So I have X and Y of this pen on the tablet, so I'm just moving it. So once I have this displacements, I plot this displacement, I have an X value, then I have this corresponding velocity profile by taking differentiation, then I'll have velocity profile with which we can say whether the person is an novel or an experienced person in a task. So this is how motor learning, quantifying the motor learning in the biomechanics framework will tell us whether the person is a learned person or experienced in the task person or having a lesser experience in the task. The same thing is applicable for typing as well. So any motor task. This is applicable for any motor task, not only for the simple teaching tasks. So another cute experiment that was performed was so let's say no, we are holding a cup of coffee. Suddenly the light goes off. Suddenly the light goes off. What happens? Suddenly the light goes off. Two things that we most of the time we might happen. One is we over squeeze the cup, or the liquid will fall on us. So in order to quantify this phenomena called visual occlusion, so my goal is to not to all the make the hot coffee pour on me or pour on my fingers or pour it on the ground. So what I do is, what we did actually is to have a experiment. So these are like four sensors. So they need to actually make a movement between these values in the. So this is how you, there will be a wedge. Will be producing normal forces. Okay, this is the sensor plate. They produce normal forces. We used 88 nanos and piezoelectric sensors. So we calculate the maximum voluntary contraction. Then we normalize the forces. So we have to produce the forces within this range. That is equivalent of holding a cup of coffee between this range. Generally, what we do, we actually switch off the feedback. They have to still maintain the instructions. Still, they need to maintain. Okay. So what we do, there are two conditions. There's a condition. One condition. There's only one condition. Simple experiment. What we did was we wanted to make sure that we give an instruction. So we'll show the performance of the next trial. So the vision is gone. Then we'll show the person how better you perform. One group. No, another group we will not show them. So the group that was having epilogue, so epilogue is with feedback and without feedback, the reduction in forces, one of the fundamental thing happens is reduction is force. This we called as force drift, unintentional force drift, because my intention is to hold the cup, but due to the lack of visual feedback, there's an unintentional force drift. So there was an unintentional force drift in people without epilogue. You can see that for all the fingers. So what this suggests is, as we previously saw, there are like a group of central controlling variables such as your L0. We call this as threshold values or resting length of your muscle. These lengths we actually select appropriately for these tasks. So within the framework, within the distribution of this task, we select them. That is the reason the central nervous system knows which control variables to select for when you show the feedback and when you don't show the feedback. So this is regarding motor control. Okay, so for learning a new task, so for learning, you always need to know. You cannot start from a existing task. Then you cannot find uh, changes in performance. Everything will be same. If a person is an expert, there won't be changes in the performance. We'll see that in the next slide. So what happened is we actually came up with a task. So we used a finger. I used a finger thumb push in movement. So this is a day-to-day movement. So you use it in pulling up of a zip, stitching. This is a movement doing almost all the day-to-day tasks. Index finger and thumb for putting salt, for actually taking pen and writing. All the tasks we do. This is a very very important task in a day-to-day activity. So given that as a context, so what I did is I wanted to characterize this finger thumb position movement. So we used a electromagnetic tracking sensor. So this tracking sensor, I need to move the finger thumb to various different positions, and based on that, the positions, how the movements are. So so this is how the characterization of the movements is. Just like how we saw with Sosnick's works, they were in line with the previous literature. So there is a shaped curve for both for different targets for the thumb and the finger. Then you will be having a, you can see velocity profile for both thumb and so the darker lines are for the thumb, and the dotted lines are for the fingers, and then you can see how the velocity profile actually changes for different this thing. What we were able to find was one of the important findings in our research work was the movements of the thumb were larger. Okay, so when I'm making a finger thumb opposition movement, what is that movement? This is that movement. This is my hand during the experimentation. This is that movement. Finger thumb is moving to different segment. This is that movement. And finger and thumb is movement. Major contribution is done by the thumb. Similarly, the movements are much smoother. Movements produced by the thumb are much smoother. So this resulted in another research work. So this shows us. So why this is happening is very simple. The both the two ways. One is peripheral. In the peripheral level, thumb is having more representations than the fingers. At the same time, thumb is having so the muscles that is controlling the movements of the fingers and the thumb are different. Thumb is having specific muscles. They have a specific set of cortical muscles below the fulcrum of the thumb, while these are actually getting muscles from the external to the hand, external to your hand. So these are like flexor digitorum profundus, superficialis, but here are brevis muscles that are actually sitting below the thumb. So they have like dedicated muscles for a single movement. That is why the movements are smoother as well as we saw also smoother movement, not a discrete movement when compared to the fingers. Okay. So after characterizing this finger thumb movement, as I told you, once you generate the movement, we need to know how a person actually becomes experts and when do they actually become an expert in this movement. Why this is very much important, very short, so we can use it for training a elite individual. By actually identifying a critical points in learning that will make them an expert. And how do we do that? We actually do something called sequence learning. So we actually sequence the set of movements, order them. So in Bharatanatyam, there are like set of sequence of movements. They ordered them so that it comes out as a that uh, a uh, meaningful ideology of the scene in a more dramatic way. So this is also sequence learning, just like how you actually chunk the phone numbers. So both finger thumb opposition movements. Plus this, we ended up in producing a motor sequence learning paradigm for learning, unlearning, and relearning. So my goal was to understand how and when people actually become an expert. So how do I did? I came up with this task. So this task, no, they need to play. So there will be specific words. They need to type these words, and key press key release information should be going into the screen. This is also published work. And so what is the goal? This is the goal. I guess this can be played. Because you can see now. Yeah, it's coming. You can see now. So the person will be actually typing the words using the glove and spaces based on whatever there is the finger thumb opposition movements, and their goal is to make this guy between these threshold values and move it on the spacer. So they will be keep on moving, and if they are typing very slow, this is the words per minute. If it's going very slow, they will crash with the building. So this game we actually reprogram this game. This game we reprogram. We purchase this game and we reprogram this game based on, because we can control the words that's coming in, and we can also change the speed. We also keep and it will also give you speed up, speed up. So this is words per minute. So if they're typing very less words and with more errors, they will crash. Else they will reach the destination. So this is the task. So this is what people want to learn. And for this, no, we had so many inventories. We recruited so many participants. Just want to know how when actually they become learn. So if they already have played this task, there's no point in conducting this experiment. And if they are very good in finger movements and they are like good in typewriting, or Cassie, we have excluded those participants because they have an added advantage, because they are, so any learning is actually generalizing from a previous task. So we don't want to give that generalization from the previous task. So we remove those kind of this thing myself who recruited them in the study, and these are the outcomes of the study. So with practice, the people try to preserve both speed and accuracy. So that is one of the important aspects, which was very much clear with the dwell time onset. Also, whenever an error comes, they still actually typed faster. So what you usually do in early stages of learning, you have an error, you don't want to make another error, so you type less as faster, right? You type with a lesser speed. But with practice, you even though you get an error, you still type with you try to preserve the speed because you know that the task pretty much well. So that is one of the reasons, and this is how you will try to have the course learning curves being fitted. So initially there will be like, so errors are reducing substantially with around like sixth day the errors saturate. So this saturation point we need to identify. That is when people actually learn a task like this, and how they actually task. They actually preserve both speed and accuracy once they become an expert. Before they become an expert, they actually try to worry more on accuracy rather than on speed. That is one of the strategies that we were able to identify with this set of experiment. This is also published work. And as I told you previously, this DMN and DAN, which we saw, actually have different fMRI implications for various different functionalities. So there's whenever there's a stroke or hemorrhage, so there is like two things: ischemic stroke, hemorrhagic stroke. One is a block, another one is due to a burst of a blood vessel. So those things could be seen here in the images. So once you have those things seen in the images during stroke after fMRI, you can clearly see they have a problem. So this is another aspect that I always wanted to work was a stroke, because we can these people they have lost the normal rhythm, but they can get the normal rhythm with appropriate practice regime. So that is why learning or finding these kind of strategies, practice regimes will enable us to actually gain access to train and improve the quality of life for people. That is the reason, and you can see one side of the body is getting affected. This will result in something called hemiplegia. So this was a funded research. Uh, I got this research funded by TNSEST. So where we were having like two gloves. So this person actually has one side of the body working, the other side of the body has failed to work, is failed. So what happens is, if you see the person. So this is actually flexor digitum profundus and superficialis. These muscles are exterior muscles. I'll just uh, there's a point. So these muscles will get a 10 milliamps of current. So based on my contraction, and this is a simulator, this is what the world is currently doing. But the problem here is ethics committee. You cannot clear the ethics committee pretty much easily if you are giving current. 10 milliamps current is actually it is a safe value. It's not going to trigger your wrath or cause any kind of a fibrillation. But still, it is very tough to get the ethics committee cleared. So you okay. So I guess you can understand. So you can see these diagrams either left and right or up and down, or those areas will be affected. Paratic, they become paralysis, they become paralytic. So the normal, and I'm not getting. So here another important aspect that people came up with is this is contralaterally control functional electrical stimulation. You're not taking anything from the brain. You're actually making some movements here, and based on that, you're stimulating these muscles. So that is one better great advantage. But what I did, so this is what existed. So what I did is very simple. So I used so based on the sensors here, I used some motor framework, exoskeletal motor. So what they need to do is, so whenever I contract the motors and the exoskeleton that is connected to this hand will be moving. So they need to practice this game, same game. So, since this game is also same, they wanted to use the same game for the purpose. So they need to because these people have cognitive imbalances. So in order to avoid this cognitive imbalances, I just changed the words to colors. So they need to just type the tip. So whenever I type the tip of the right hand, the paratic hand also will make a move because they are connected with motor exoskeleton. So this was funded by TNSCST, and we completed this project. And we assessed the stroke patient motor assessment using this box ball. So once after doing this, they have to pick and place using the affected hand from this point to this point. We'll have a score. Similarly, they need to interact with these things from inside to outside. Then we will put the Fulger assessment score, and based on that assessment score improve. So that was the project all about. So with this, I have concluded my talk. It's been an elaborate talk, but these are the takeaway messages which I feel to be emphasized at the end of this presentation. So one is we saw today about various new research areas in the field of biomedical engineering. To begin with, neuroscience already existing. So brain and spinal circuits. How both of these circuits are formed to perform a specific task. So whenever you go for an environment or interact with the environment task, you immediately jump to control, because that is where you will be planning, initiating, regulation, generation of movements before the actual movements. So this actually takes place in the back side of the screen, in the central nervous system before the actual movements are actually generated. So before, for generating these movements, you need some circuits, you need some connections. Those are studied by neuroscience. Then once the actual goal directed movements are motor action. So these goal directed movements cannot be accurate most of the time. So what we do, we actually give them the practice with feedback, which results in less time and effort to achieve the outcome. And this is where the skill and behavior now comes into play. So once you are able to do a task in a repeated way with less time and effort, you make it as a set of similar movement patterns. Okay, these similar movement patterns actually became a very good outcome in a equivalent solution. They become a very good outcome. They will be stored as a skill, and the behavior that is going to accomplish your task successfully is called a skill. And this behavior will always result in permanent change in neural structures such as memories. So this skill you are is stored in the memory, and you are retrieving the skill to accomplish the outcome of a task. As I told you, driving, driven for 10 lakh kilometers, it's stored in my permanent memory, because from working memory it shifts to permanent memory. And after certain period in time, also I'm able to retrieve the important features to execute that motor task successfully. So that is called a skill. Then using mechanics such as force and geometry. such as kinetics and kinematics to quantify human movement is called biomechanics. So that is a behavioral aspect. That is the outcome-based aspect. But in order to understand how the master or the loose controller or CNS is working, we go for fMRI for physiological aspect of neuronal circuits, which is getting activated, which region is getting activated. So, for example, we saw that one of the regions after stroke is getting affected. Is that region getting activated one more time? Is that any functional? Is there any activation happening in the affected region, or is there? So most of the time with physiotherapy and with the rigorous training session, the regions next to the affected stroke area will take over the functionality. Is it actually happening? Those things we can see using fMRI. Okay, so these things can be used to improve our tasks in day-to-day activity. So these are certain takeaway messages, and these are my active areas of research where I actually quantify movements in real-time environments for normal, for athletes, as well as for neurologically developmental disorder kids such as autistic kids. So that is so where we are developing an AR-based system for autistic kids for their to improve their uh, to actually reduce the stress on the parents, to make them more and more again becoming more performance oriented child. Then more other researches on fMRI, Alzheimer, Parkinson's disease, immunotherapy. So my goal is actually to combine the fMRI and the behavioral for a day-to-day activity task. In the practice, I'm also actually developing such a electrodes for ECG. So this one electrodes and this one of these three things are my all these four things are my scholar works. I have like 2 to 4 scholars. Two scholars I guide and two scholars I am co-guiding them. So all these are areas that I'm presently actively working. And if you have any questions, you can ask me now. And these are my contact details. You can also contact me on any kind of discussion regarding any small help regarding your work or anything that is going to that I could do from my side. I'm always happy to do. And these are my contact details. So any queries? I know this has got taken a long time, but uh, I thought this will be pretty much important for everybody to know that how new areas of biomedical have evolved and why they are very much important. I really thank everybody for know, being with me through this session. I thank the organizers. I thank Dr. Gauri, madam, for giving me this opportunity. She came up with this idea. We can do this. So it is always a responsibility to share the information that we have, very small information that they have with our colleagues. So I thank the WITDM and Mahavita teaching program hosted in WITDM for giving me this opportunity. Thank you. Any questions you can always reach me to these things. I'm always open to have live discussions. Madam, hello.
Dear participants, if there are any queries, you can ask.
Hi, Dhanush sir. New Year.
Tell me, sir.
The thing is, you were concentrating on fMRI in terms of quantitative analysis or qualitative analysis.
Quantitative, sir. Qualitative, we are actually not doing. We are doing at the screening level, sir. But the quantitative only we are going. We are actually developing models, and we are statistically supporting. We are developing statistical frameworks to support our claims, sir. So quantitative analysis we are going, sir.
Okay, Dhanush sir. And one more question. Is there any specific terms in terms of focusing on white matter, gray matter, or CFS in
Yes,
terms
sir.
of
Actually, yes, sir. Yes, sir. Sir, actually, I forgot to mention what we another important term in schizophrenia patients. What we do is we actually calculate the volume of gray matter and white matter, sir. So the difference in this volumes actually are different for different. It is actually different for people with AVH and without AVH, audio verbal hallucination. So as you rightly pointed out, that is actually a very important aspect, sir. You told that, so that number we are actually we are actually calculating, and we are using that to differentiate. And what we wanted to showcase was it was different for both hallucinating and non-hallucinating, but that was not the case. So then we need to go for other metrics to actually differentiate them, sir. Yes,
okay, okay, Dhanush sir. Thank you for your reply. We will catch you up. We'll
definitely,
catch
sir.
you. Up.
Madam. Madam.
Sir, if there are no queries, can we wind up the session?
Yes, madam.
Yeah, okay. So I extend my heartfelt thanks to our speaker, Dr. Danush, for delivering such an insightful and enriching session. Your expertise and clarity have greatly added to today's program. Thank you, sir, for sharing your time, knowledge, and experience with us. We truly appreciate your contribution. Thank you once again, sir.
Thank you, madam. Thanks for giving me this opportunity. Thank you.
Thank you, sir. So, dear participants, we will have a short break and we look forward to giving you the same strength by attending the next session at 2 p.m. So, I kindly request all the participants join the meeting before 10 minutes. Thank you all.